Nipah virus was first emerged in 1998 following an outbreak among Malaysian pig farmers. There were no outbreaks recorded in Malaysia since 1999, but there were multiple outbreaks recorded in Bangladesh and India, along with several deaths. The Nipah virus was named after a village in Malaysia, Kampung Sungai Nipah in Negeri Sembilan. The animal host reservoir for the virus is the fruit bat, which is commonly referred to as the flying fox. The fatality rate of this virus is estimated to be between 40% and 75%, which led to roughly 300 human cases, over 100 fatalities, and significant economic effects in Malaysia. Most human infections during the first known outbreak in Malaysia happened as a result of encountering sick pig's body fluid or the contaminated tissues. The Nipah virus is not an airborne disease. Instead, it is transmitted through body fluids such as coughing and sneezing. Till now, no vaccine is safe and effective for human use to cure this infection. The Nipah virus belongs to the genus Nipah virus in the family of Paramyxoviridae. It is a rare zoonosis that primarily spreads between animals and humans, which may cause severe illnesses. Moreover, it is an envelope virus with spikes of glycoproteins that extend from the viral envelope. Besides that, the Nipah virus has an alpha helical capsid. The capsid plays an important role for the attachment of the virus. It also contains a negative sense, single-stranded, and non-segmented viral RNA. Due to this, the Nipah virus belongs to the class 5 in the Baltimore classification of viruses.
Pathogenicity refers to a pathogen's ability to cause disease. Fruit bats, often called flying foxes, are where the Nipah virus started. The fruits that the bat chews on are infected with the infectious saliva and urine, and when the pigs consume them, pigs become the most infectious in Malaysia compared to flying foxes. There are two types of transmission, direct and indirect. As for Nipah virus, it spreads directly by droplet spread and direct contact. Direct contact with infected pigs or bats, including their body fluid, can cause infection in humans. In addition, if a person is in close contact with an infected individual who coughs or sneezes nearby, droplet transmission may occur. A virus is an cellular particle that needs a host in order to reproduce. An envelope virus, the Nipah virus penetrates its host cells by direct fusion. As soon as the virus surface, which was coated with glycoprotein, made contact with the host cell's receptor, the infection began. UV2 and actin B3 are used by the Nipah virus in this instance as entrance receptor. The viral envelope then interacts with the host cell membrane through the fusion protein, allowing the nucleocapsid to enter. The nucleocapsid will then subsequently uncoated, allowing the RNA virus to replicate in the cytoplasm. The virus is a negative sense RNA virus. Thus, before it can be synthesized, it must be converted into positive sense RNA called mRNA. The virion is fully mature, matrix protein assists in its assembly and binding. To prepare the plasma membrane for the release of the virus, the matrix protein organizes the assembly of cytoplasmic nucleocapsids and surface glycoproteins. The nucleocapsid will attach to the membrane, forming a small pouch around the virion. The pouch is pinched to release the new virion together with its envelope.
Nipah virus is a zoonotic pathogen that can be transmitted from animals to humans. Thus, this virus can cause mild to severe disease that impact your human health. The initial signs and symptoms of Nipah virus infections are non-specific because symptoms typically appear in 4 to 14 days following exposure to the virus. Symptoms may initially include one or several of the following, such as headache, cough, sore throats, difficulty breathing, and vomiting. These can be followed by the dizziness, drowsiness, and neurological signs that indicate encephalitis, which is the severe symptoms of this virus. Encephalitis is an uncommon but serious condition in which the brain becomes inflamed. It can be life-threatening and requires urgent treatment in hospital. Anyone can be affected, but the very young and very old are most risk of this disease. Besides, some people can also experience atypical pneumonia and severe respiratory problems including respiratory distress. Encephalitis and seizures occur in severe cases progressing to coma within 24 to 48 hours. Most people who survive encephalitis make a full recovery, but long-term neurologic conditions have been reported in survivors. Approximately 30% of patients are left with residual neurological consequences such as seizure, disorder, and personality changes.
Okay, now I want to talk about impact to human in economy. Apart from the human catastrophe of the high morbidity and mortality rates during the epidemic outbreak, the economic impact of the Nipah virus is tremendous. In 1998, Bukit Pelanduk had a major socio-economic impact on the life of the patients, their families as well as the whole village in general. The living standards of most affected families had been reduced in the long term, not due to medical expenditures, but mainly due to loss of income and employment on the pig farm. After the Nipah virus outbreak in 1999, the Malaysian pig industry and related sectors suffered enormous damage. 1.1 million pigs were killed, which a total decrease in the Malaysian economy of around 30% during that time. This required the additional $136 million cost for a control program for biosecurity and slaughter facilities. In addition to direct loss in the livestock sector, the feed industry and oil and fat production were most affected.
So now I would like to discuss about how the government control when the Nipah virus spreading. One of the first approach by government is by control measures such as mosquito fogging and also vaccination of pigs against JE were deployed to the affected area. However, it was still ineffective since number of cases still increasing. Healthcare workers who were taking care of patients that were infected had been convinced since the outbreak was not caused by JE. Further autopsies were inconsistent and they suggested that it may come from another agent. It was supported by the fact that all of the infected people had direct contact with pigs, and all of the pigs that had been infected had severe symptoms of barking cough before dying. Despite the evidence gathered, federal government, especially health authorities, stated that JE was the main cause. After the findings, widespread surveillance of pig populations together with extermination of over 1 million pigs was undertaken, and the last human fatality occurred on 27th May 1999. The outbreak in Singapore also ended by the prohibition of pigs importation to the country immediately and their subsequent closure of slaughterhouses.
One of the interesting facts about the Nipah virus is that the outbreak of the virus was initially mistaken for Japanese encephalitis, but local doctors noticed that adults made up an unusually high percentage of the cases, and that people who had received the Japanese encephalitis vaccine were not protected against the epidemic. Even though these findings were made within the first month of the outbreak, the Ministry of Health overlooked them when a national campaign was started to inform people about the risk posed by Culex mosquitoes, the mosquito species that transmits Japanese encephalitis. Other than that, do you know that fruit bats of the genus Theropus, which is flying foxes, are the main reservoir hosts for Nipah virus? P. vampirus, the Malayan flying fox, and P. hypomelanus, the island flying fox, are known to carry this virus in Malaysia. Gigantes is thought to be an important host in Bangladesh and India, and possibly other locations. Although live virus has not yet been isolated from the species, Nipah virus RNA has been detected, and many bats are seropositive. Not only that, this virus can survive for up to 3 days in some fruit juices or mango fruit, and for at least 7 days in artificial date palm sap that held at 22 degrees Celsius. This virus is reported to have a half-life of 18 hours in the urine of fruit bats.